Awesome. All right. So, all right. So we've got 15 hives outside Boise. All right. So we're going to get started today. I'm just going to check Nicole and Peter. Is my sound okay?
Sounds great.
Yep. Okay.
Yes.
All right.
Sounds very good.
Fantastic. So welcome everyone. We're going to be talking about bee vectoring today. This might be an entirely new topic for you, but Dr. Kevan has actually been studying this. How many years, Peter, have you been studying vectoring, apivectoring?
We started working on apivectoring back in the late 1980s. So we've been doing it a long time. So it's developed quite well, I think, and really with all the new and available biological control agents and the dispensers and everything else, it's really come along very, very fast in the last 3, 4, 5 years, I would say.
Yeah. So anyways, I've been very privileged to be working with Dr. Kevan for the last couple of years, and we have a number of research assistants who have been helping out with our research. So Nicole's joining us today. She is a master's student at the University of Waterloo, and she also is helps out with our research and our beekeeping stuff. And at the end of our session today, we're going to be taking questions, and Nicole will be passing those along to us so that we can make sure we get through the questions that we can today. And Dr. Kevan will be staying on with us as well. So when we get to the end of our presentation, if you have any questions that are directed towards him, you can go ahead and ask him as well. So I'm going to get started. I'm going to share my screen now. All right. And all right. So
We see your presenter mode, Erica, if you wanna.
Okay. I don't know how to.
So if you go display settings, click on that and swap presenter. There you go.
All right. What do you see right now?
Currently it's a black screen.
Okay, that's all I see.
There we go.
Okay, great. It was going to be a very hard presentation with a black screen. All right. Okay, so welcome. I'm Erica Shelley, Dr. Erica Shelley, CEO and founder of Best for Bees. Today we're talking about bee vectoring to improve honeybee health. You'll see my contact information there, my company's contact information. So if you have any questions that we don't cover at the end of the presentation, or if you have to leave the presentation early today, you can contact us on any of those places to reach out and connect with us. We are recording the presentation today, and you will find it on our website, bestforbees.com, as well as we'll post it on our Facebook page and Twitter and like any other social media things that we have. So to get started, a little bit about myself. I've had this company, Best for Bees, for several years. Over 10 years, I've been working as a job with bees. So just like all of you that probably got into beekeeping, or maybe even are wanting to get into beekeeping, you know, I was fascinated with the insects, and I was fascinated with sort of all the social things that happen inside a hive. But the truth is, I was really, really wanting honey. And it was just the honey that got me into it. And little did I know where my love for honey was going to take me. And it has been an exceptional journey. So a little bit about some of the things that we do at Best for Bees. We used to do more in terms of local things before we got into more research. but we've always had like an education component to Best for Bees. So we had an observation hive that we would take out to events or into schools to teach kids about what happens inside hives. We had beehives that are at different companies that allow us to educate the employees about the importance of bees, what they are to our food system, how to provide pollinator habitat. And all of these are often part of sort of green initiatives that a lot of companies have to work with. Additionally, we've worked with our local library, the Kitchener Public Library. So a little bit about where I am. I'm in a town or a city called Kitchener, Waterloo. It's two cities that are right together. And we're about an hour outside of Toronto, Ontario, Canada, and about 30 minutes from the University of Guelph, which is where Dr. Kevan has been doing his research for years and years and years. And so the Kitchener Public Library, we put in some hives there, which are great because if you look at that, you'll see that there's just these windows they're right at eye level, so people can see what's happening with the bees. And they had a little bit of a scare last week. We had a warm day after a long cold spell, and there were tons of dead bees and poop all over, and people were just really worried about the bees. And we're like, that's a great sign. They're cleaning out their hives and they're doing all the things they haven't been able to do because the weather has been cold here. Another thing that we do is we work in the community in various ways. So sometimes people get bees moving into their homes, and we go in and we will remove hives from homes or buildings. We've done high rises before, so the bees don't care. You know, they find a nice, warm, perfectly sized space, and they'll move in there. So sometimes we'll take those out. We have Bee City Kitchener, which is there's bee cities in Canada and bee cities in the United States, and the mandates of being a bee city are basically to educate the people that live in those cities about the importance of pollinators, how to provide pollinator habitat, and you know what, just overall understanding that not, you know, our food resources, but also just all of our plants and everything are dependent on pollinators. And the byproduct, of course, of beekeeping in any sense is that I am left with some honey. And so we get to sell honey and candles and beeswax wraps and do those kinds of things depending on the time that we have to do them. So yeah, so we kind of are normal beekeepers that way. But then we have a whole other side to what we do, and that's the research side of bees. So my background is I have my PhD in molecular and medical genetics, and for years I was a beekeeper, and my science did not cross over with beekeeping. But then there came a point where I realized how much I loved bees, and the more that I had been doing work, I'd started just doing basic science research, and a lot of opportunities started coming my way that allowed me to start doing research with bees. And it was just this great intersection between the microbiology background that I had and that love for bees. And so started doing research with bumblebees and honeybees. And some of that research is in laboratories. So, you know, still, you know, doing things like PCR and plating plates and all the kind of good stuff that you would do inside a lab. But other times are out at farms and working in our apiaries and getting samples there. So just honestly, one of the best jobs I could ever have, that I get to be outside a ton working with bees, but also doing the science stuff. And over on the far right here, you'll see some of our research assistants from this last year. So this is Tasman Brown and Aparna Karthikeyan and myself in front of our apiary from last year. So just a few members of our team, lots of people working on different levels to do different things. And in the center here, this is a bumblebee that it was a memory study that we were doing as well. So lots of different things going on. But it was this research component that connected us with Dr. Kevan at the University of Guelph. He had been studying this process called apivectoring or entomovectoring for insects carrying something, apivectoring, and what we're now calling bee vectoring. It's a little easier for people to understand those words. So Peter, this is him in front of some hives here. As I mentioned, he's a professor emeritus at the University of Guelph. I think he has a couple of bees named after him. He's got publications in the several hundreds of publications, and is really just I just want to say just super esteemed in the field of bee research and plant research as well. He's worked in botany stuff as well. So as he mentioned, late 90s, he started working on this idea of bee vectoring. And he worked with some other scientists, John Sutton and Les Ship, to develop biocontrol methods to combat pests on crops. And I know that's a lot of big words, but biocontrol just means where you use something that's biological, right? And it's usually like a natural thing, something you would find in the environment. And they then would use those things to fight any pests that were on crops was the idea behind it. Now, bee vectoring is actually super simple concept. And the idea is that a bee walks through a powder and that powder sticks on that bee. And then that bee goes on its normal pollination visits from flower to flower to flower. And as it visits, it leaves little traces of that powder behind. Now, the original powders that they were using were soil fungi. So the soil fungi would be left behind, and then it would grow, and it took up space, so it can actually keep other fungi from going, but it also is an entomopathogenic fungi, which entomo, again, we're coming back, that means insect, and pathogen means it's going to kill that insect. So big words, but very simple in terms of that it basically gets into the insect and kills the insect, and actually grows on it, and just, it's just it's a growth medium at that point, that insect. So what you'll see in these pictures here, these are not honeybees at all. These are bumblebees. And in fact, the bee vectoring originally was done mostly with bumblebees. And there's several reasons that was done. One of them is because bumblebees will fly in a lot cooler weather than honeybees. And so a lot of crops are flowering earlier in the season. And so you actually need bees that are flying at that time of year. And so bumblebees are ideal. They don't care if it's cooler, they're also really effective pollinators. So they can do lots of trips, and they're big, so they can carry lots of powder, and they're not as clean as honeybees. So if you put a powder into a honeybee colony, they're more likely to remove it quickly and not necessarily carry it. Whereas bumblebees are a little bit less cleaning up of their hives, and so they'll tend to keep the powders there. So bee vectoring, it's really simple. We say using managed pollinators. And so managed pollinators can be mason bees, bumblebees, honeybees, to deliver beneficial agents. So Peter, of course, he pioneered this, and he actually has worked with companies, and they have patents, and they are now delivering this fungi out to crops such as strawberries and blueberries and even almonds, to protect the crops against pests and diseases. So he had the idea, and I have no idea, Peter, when you came up with the idea of what we call the Inspencing Project, this idea of bees carrying powders into the hive to protect bees from pests and diseases. So yeah, so I got a little picture of Peter over here, and it says, could bee vectoring be used inside the hive? You like that, Peter, with the little bubble above your head? So he had previously had a student that had developed a prototype for delivery, and he came to Best for Bees and asked if we would be interested in joining him in this project, which of course we were. And so we jointly applied and received funding for a 2-year project, which was called the Inspencing Project. Now, let me describe what inspencing actually means. Inspencing is the idea of powder being taken into a hive, whereas outspencing is the powder going out. So we just had to differentiate between bee vectoring going out and bee vectoring going in. So these are Peter's words, Dr. Kevan's words, outspencing and inspencing. And so, yes, we worked on this inspencing project for 2 years. And in fact, the device that we were working with, we originally called the Inspencer. Now, why even bother with bee vectoring? Well, it's easy. It is so easy to like just throw a powder at the front of a hive and let the bees do the work for you. But there's other advantages as well. You don't have to take apart the hive to put medications or treatments into the hive. Every time you take a hive apart, you're disrupting the entire ecosystem inside a hive. And don't get me wrong, I'm not saying don't ever go into your hive and do your inspections. It is important to do all of that. But the less that you have to do it, the better. If you're dealing with a small hive beetle, sometimes they're put in little propolis gels. And so like if you disrupt it, you're actually, letting all those small hive beetles kind of run out. The queen will quit laying for a few days. You can smush bees, they get aggravated when you go in there. So this is one less time that you would have to go in there if you could just put a powder at the front of the hive. Additionally, if it's raining, if it's cold, if it's evening, not a problem because you don't have to open up the hive. You could potentially add multivitamins or multiple doses of things over and over again, which would allow you to, stay on top of any treatments that you might need to do for your bees. And you might only need a few devices per apiary if you move your bee vectoring devices around. And as I mentioned, it's fast. When we do our bee vectoring, it takes us less than a minute to actually add the powder. Within 6 hours, the bees have emptied it, and it's done. So that's a pretty fantastic advantage as well. So as we were going into this, I had to think about, all right, what are all the requirements? Dr. Kevan's lab and his student had thought about these things in the past, but there were a couple of things that were missed, or maybe not missed, but weren't a concern early on that we realized needed to be addressed. So the first thing is it needed to stick to the bees. So there's a powder that we call the diluent that is bee friendly, and it sticks to the bees, but not too much. You don't want something that sticks so much that it doesn't fall off. So it has this magical place where it sticks enough to the bees, but also falls off as needed. Also, it needs to not be toxic to the bees. So when we're looking at toxicity, we're actually looking at a couple of different things. The first thing we're looking at is obviously can the substance that we're adding to it, make them sick. But we also have to look at the size as well. Anything that's too small can actually affect their breathing. So we have to pay attention to the size of the particles as well. The next part, a little bit harder, would need to make sure bees didn't move it out. So for any of those of you that might already be keeping bees, or if you've and if you've ever done a removal from a structure where you've used string or anything like that, the bees will like yank the string out within a couple of days. Like they do not want anything in their hive that doesn't belong there. They're super clean. So outsmarting the bees to keep them from moving it out. We didn't realize that was going to be important initially, but when there's a lot of waste happening, you don't know what's going out as much. And if you specifically need certain concentrations of whatever you're adding to go into the hive, that's an important aspect to consider. And then could the bees carry it throughout the hive? What if they like take it in and they just drop it off like right in the very first area that they go to? So we needed to make sure that it could actually be distributed throughout the entire hive. And then how could we tell that it was actually getting distributed throughout the hive? And I've got a little hint in the picture over here. So you see a little hint, and it's pointing at a little pink substance on the back of this bee that's sneaking into the hive. So what we did is we took microbeads, fluorescent microbeads, and we mixed them with the diluent. And you'll notice here there's also little granules of rice, and that's because this can get wet, right? So we really wanted to make sure we kept it dry. So as I mentioned, Dr. Kevan's team had previously developed that diluent that easily stuck to the bees without harming them. So that was very easy for us. We added the fluorescent powder, and then we added rice to keep it dry. And then what we would do is we would take samples of a single cell or 3 cells, and we would suspend those cells in a liquid and then look at the most minute amount, 10 microliters, under a microscope and see if we saw any fluorescent particles. And what we did see was actually very, very positive. So I'm going to first of all show you our research apiary here. So this is our first year. This is Tasman Brown. So she was one of our research assistants, and we set up our original 12 hives. This is at Strome's Farm in Guelph, Ontario. Strome's Farm, they have graciously had us at their farm for many years, and we are very, very thankful that not only do they allow us to be there when they're considering different things to use on their crops, they always check in and see if it's a bee friendly thing as well. So they very much, it's great partnership to be working with them. And if you look over here on the left, this is us installing our nukes. So it's important when we do these studies that basically the bees are equalized throughout all the hives, and that basically everything's the same, which anybody who's worked with nukes, when you get nukes, sometimes there's more bees and less bees in a nuke. And let me rewind. What is a nuke? If you don't know what a nuke is, a nucleus hive is basically 4 frames of bees that you get to start a hive, and it'll have a queen. It'll have bees that are taking care of the babies, what we call nurse bees, and there'll be babies in there, which we call brood. And then there will also be worker bees. So we have that. And then we also will have a frame of honey and some pollen that will be food sources as well. So we have 4 or 5, depending on where you are, frames that go into the hives. And then we put other frames into those hives to fill it out. And then over the year, it'll get bigger and bigger as they fill it up with honey. So our Inspencer that we were using, the one that had been developed, was this tray Inspencer, and it was basically a big sheet that was the size of the bottom board where the bees enter in, and it had all these ramps that the bees could go up in. So we had 12 and an 8 sized ramps, and we were going to compare those to see which one worked better for bringing in powders to the hive. It basically had 2 levels. So if you look at where the number 8 is there, on the bottom level, you can see that pink diluent that we, and the fluorescent beads that we had put in there. And that would be where they would be going in, is on the bottom, and then they would go up those ramps and then into the hive. And then the idea was then on the second level And you'll notice there's a piece of wood that goes around the outside of it so that you basically have one bottom board on top of another. Would be the side, the level that the bees would be flying out. So there would be an in and an out. And that way the bees would just be carrying the powder in and not carrying it out. It did not work. So we tried all sorts of ideas to get the bees to go in and out the way that we were hoping they would. And we had a 2-year grant based on this, and it just was not working for us in the field. The bees were moving all of the powder out, and we were a little worried about them carrying, you know, in the future, depending on what we put in there, them carrying tons of that out of the hive. So we got creative. We started thinking of different ways that we could convince the bees to go in on the different levels. We even, this right here represents like when you've just gotten so desperate with your science that you're trying to think of other ideas of something that can happen. So we did little coloring sheets and patterns that we had read might work well with getting the bees to go in, and it didn't work. So if you look at our graph here, the blue represents what percentage of bees are going in where they should be going in. The yellow represents how many bees are going, what percentage of bees are going out where they should be going out. And then the red is overall how many are doing the right thing, going in the indoor and out the outdoor. And whether it was the 8 ramp tray or the 14 ramp tray, it was about 50%, which just basically means the bees were going in and out wherever they wanted to go. So, you know, going in through the outdoor, if you know the Prince song, she went in through the outdoor, not great for our research here. So we thought we need to come up with something new, something different. And I started looking around, asking on beekeeping forums and beekeeping groups, people ideas, and trust me, every idea that you've ever heard came our way of ideas of getting this to work. And then one day, I just woke up 3 a.m. and I was like, cones, the cones that are used on bee escapes. I could use those alternating and have an in and an outdoor. I could have 2 chambers, and then the bees would carry the powder in or carry it out. And luckily I had a friend who was doing a huge renovation project and had a little bit of extra wood kicking around because wood was really, really hard to find. It was in the middle of COVID. And so we kind of pieced these together. Look at the beautiful duct tape work here. So I'm just going to show you one of our first times of doing this. I added component dispenser number 5. Gathering along this edge here, which they tend to do. Going in, going right in, nice and easy. No problem. On the other side. Is that video working for everyone? Yeah, okay. All right, and so that was basically the first time we were getting them onto the hives. You'll notice there is a gap at the back, and so anytime there's a gap, the bees will try to go in and they'll land there, but they figured it out usually within a half hour or less of us putting it onto a hive. They'd figured the in and the out. And then we just started our studies then with the powders to see how things would go. And did that do the trick? Absolutely. So if you look, this is our control. It's, you know, just open. So everybody's flying in and out wherever they want, which is fine. And I already showed you our results at about 50-50 with the 2 different types of trays. But with our cones, the in, at least 80% were going in where they should be going in, and over 90% out where they should be going out. And overall, the average was roughly around 85%, which was a great improvement over what had happened before. And so now we were able to look at whether the bees were tracking the powder through the hive. And as I mentioned, we would take those three cells, suspend them. We did different quadrants. We did alternating frames. We would check the supers, and then we would look for the fluorescent particles under a microscope. And what we found is that 80%, oops, did not mean to do that. 80% of our cells would have some fluorescent powder present. And that 80% was a very underestimated, because as I mentioned, we would take 3 cells, we would spend it in about 1,000 milliliters of water, and then we'd only take 10 microliters of that and look at that under a microscope. So so it would definitely be higher, but we definitely had managed to get the powder throughout the hive. Now, what you'll be noticing, though, you might be noticing, is that the trays actually work just fine too. They did get the powder, or the ramps definitely got the powder through, but we had a lot more powder loss, and we had to add more to those to get those effects. When we took a closer look at where the fluorescent powder was, we found that there were slight differences depending on where the frames were in the hive and in which quadrant. So if we took a frame and we divided it into 4, with the farthest away from the entrances being A and B, and the closest one C and D, and C being closest to where the bees would be coming in, you'll see that just above that quadrant one, the graph there, it says quadrant, you'll see that C, which is the closest, actually had the highest percentage, so closer to 90 to 100%, and then A a little bit higher, and then B and D a little bit lower. Still all of them over 60% or higher, but definitely some differences depending on where it was relative to the entrance and where the bees were vectoring the powders in. So first year, exciting, because we were able to prove, just like Dr. Kevan had predicted, that bees could vector powders into a hive. And this is a new method for us to potentially bring things into a beehive to combat pests and diseases. So year 2 is where things got super exciting. And that's because now we were able to test different things inside the hive to see, 1, if they could be vectored by the bees, and 2, if they could have an effect on outcomes regarding pests and diseases in the hive. So our research apiary, you'll notice, is a little bit different. So we had a couple of things that were not great with our first research apiary. So we aimed for sort of some semicircles. We had more hives this year, and then kind of just a little bit better location. We had to move the location because the wagon ride that happens at Strome's farm went right by our bees. So we moved it so it wasn't where the wagon ride was happening. And so you'll see here, we've got, at this point, we've now moved away from wood, and we've started 3D printing our ProtectaBEEs. We're playing around with different colors, different configurations. We have a lot of different tests going on to find just the right, I call it ProtectaBEE because that's what we now have named it, but at that point it was just the Inspencing. We have lots of different ones that we're testing, and then we're testing different powders to see whether they can be vectored through the hive or not. So on the far right there, you'll see we would basically measure out what we were testing with. So in this case, it was an antibiotic that we were using, and we would just spread it equally. And then we would time how long it took for the bees to empty out the trays, if there was anything left. And then usually within 24 hours afterwards, we would take our samples to see if the substances had been vectored. For ease, I should say, the first thing we decided to test was antibiotics in the hive. So a lot of people don't use antibiotics in their hives, but Here in Ontario, we have a disease called American foul brood, and American foul brood can be very dangerous to a hive, and it can spread. And in fact, if you get American foul brood in your hive here, they recommend burning your hive and all of its components. So it's a pretty serious disease. So the recommendation here is in the spring to take your hives and treat them with antibiotics, what we call prophylactic treatment. So that means ahead of time before anything bad has happened, to do a prophylactic treatment of your hive with an antibiotic. And that's because if American foul brood spores are there, if you treat with the antibiotic, it kills them before it becomes active. And so it was very easy for us to test antibiotics because you can see the presence of an antibiotic because it kills bacteria. So very easy test for us to do. So we did conventional treatment, which is where you just take powdered sugar with the antibiotic Oxytet or Oxycell, and you sprinkle it over the brood frames. And then the bees just drag that out over the brood frames. And then we also put it into our Inspencing, the ProtectaBEE, with our diluent, and then tested again 24 hours. So we compared conventional treatment to bee vectored treatment. And I have here just what we originally thought we were going to do was these little tests where you basically put a little bit of extract onto a disc, and then if it has antibiotic in it, you get this nice circle, and you can measure these circles to determine the concentration of your antibiotic. But apparently we had access to a lot of high-tech stuff, which is great. We have sort of a great relationship with Dr. Kirsi Gara at the University of Guelph, who has some things that made it really easy for us. So what we could do is just basically take bacteria and add our extracts from either our larvae or our comb to bacteria, and if there was antibiotics present, then it would kill off the bacteria and it would become clear. So we've got some tubes here just to kind of show you. If you look at the far right one, you can see how cloudy it is. But if you look at the first one or the third one from the right, you can kind of see that they're clearer. And that would indicate that there was something in there killing those bacteria. But usually we wouldn't see that. We would just put them in a 96 well plate and then slide it into a reader. And then it would just come back with exactly how much bacteria is in there, which is very, makes it really, really easy. And the best news was that we got pretty much the same amount of antibiotic presence in all our samples using bee vectoring as we did with the conventional application. So this meant that this could completely could be a substitute for conventional treatments. Here in Ontario, in the spring, it's cold, it's raining a lot. Sometimes it's hard to get into your hives. And this way you could just put the powders right in the front of the hive, and it would allow you to continue your treatments and do what you needed to do. So this now got us moving on to what is a really big threat for bees all around the world, and that is Varroa destructor, the Varroa mites. So the Varroa mites, they feed on the fat tissue of larvae, and that while they're doing that, they can actually transmit all sorts of diseases, weakening bees, weakening hives, and pretty much is one of the biggest predictors to whether your hive will die over a year or not. Up to 50% of beehives in the United States die every single year, and for lots of reasons, but the majority of those being these Varroa mites. And so in order to do some tests with the Varroa mites to see if we could actually vector powders in that would help fight the Varroa mites, we had to get our Varroa mite numbers up. So who like purposely is putting up their Varroa mite numbers? Not everybody, but we got some high counts of Varroa mites. I don't think I'll ever be proud to get high counts of Varroa mite numbers again, but we did. And so what we did is we started testing different candidates for killing Varroa mites. So there's two ways to test Varroa mite presence and numbers in a colony. The first one is using an alcohol wash. And an alcohol wash is very exact. You're counting 300 bees and you're washing them with alcohol and you're seeing how many mites come off. So that's the most accurate way to do it. But another way that people will look is at a Varroa mite drops, and they have a sticky board underneath. And as the bees die, or as the Varroa mites die, they fall down onto the sticky board. And so we did both of those methods for looking at our Varroa mite counts. But what we were really interested in looking at was whether those fungi that were being used to kill pests and diseases on crops could actually be used to control Varroa mites. And so the fungi, this is really cool. What they do is they get on top of these pests, and they use two methods to try to invade the pests. So the first one is a mechanical action. And then the second one is an enzymatic action. And all those things allow them to kind of just dig right into the pest. And then once it gets in, it starts replicating. And it basically takes over the entire host, and the host dies, and it just starts coming out of it. And it's spores, right? So the spores are actually going to keep spreading as well. And so the thinking was that we could use this fungi to control Varroa mites. And in fact, there had been multiple studies done showing that two fungi, Bavaria bassiana and Metahesium, were able to kill Varroa mites. So we thought we could use those with our bee vectoring system to control Varroa mites. And the nice thing about these fungi is that they're already present in pretty much the entire North America. You'll find these two fungi growing in the ground. So they're waiting for ground pests, dirt pests to grow on as their substrates. So it's really nice because it's natural, but it's also native to the environment. So, and also effective, like it keeps growing over and over again. It's not just a one-time thing, like it's just going to continue growing. So lots of advantages to using these fungi. Now, we originally were going to use something called Met52, and that's a Metahesium, as well as Botanigarb, which both were already used for crops, for bee vectoring, and also just spraying by farmers for crops. So these were two candidates that we were looking at using, and we were choosing to use already existing strains that had been used previously, because it would just require a change of label use instead of having to register an entirely new strain with the government agencies that regulate these kinds of things in the different countries. As we were gearing up for this second year study, a paper came out from Washington State University about Metahesium being used to control Varroa mites in honeybee colonies, but not just any Metahesium. These people had used directed evolution. So they had provided nutritional stress to the fungus, as well as other types of stress to accelerate mutations that would allow the fungus to grow inside a hive. And so because they were able to direct this evolution, they were able to choose a strain of Metahesium that would grow in the heat and humidity inside a hive. Whereas the ones that we were using, they were just being used on crops outdoors. So they weren't necessarily ideal for using inside a hive. And once we saw that paper come out, we realized that our Beauveria bassiana probably was not going to be super effective at controlling Varroa mites, but we could track it with bee vectoring. And if we found that it was being moved through the hive, that meant that we would be able to use another fungus, such as this Metahesium, to control Varroa mites inside the hive. And so the way that we measured the presence of the fungi inside the hive is we would cut out either larvae, we'd take larvae, we would take mites that had fallen and rinse them off, or we would actually look at comb and we would PCR this. And PCR is just basically where you're amplifying the DNA. So people know a lot more about PCR with COVID than they ever did before, but this is what we used. And so we found that over 50% of the larvae and around 50% of the cut comb samples had Beauveria bassiana after we had used the bee vectoring. So we tested 24 hours. And we did some 2-week and 4-week studies as well. We found some Beauveria bassiana after 2 weeks, but no, not any further after that. So it wasn't staying alive in the hive, which is really would be ideal. And we still had lots of mites. So, and in fact, if you look at the graph on the far right, you'll notice that the no treatment, the mite counts, so these are our alcohol wash counts, were close to 0 when we started. 2 weeks later, up at 20 for the no treatment, and at about, or not 20, like about 15, and then around 55 or so at 4 weeks. Whereas the one with the Beauveria bassiana, it was a little bit higher. So I wouldn't take from that the Beauveria caused the mite numbers to increase, but rather it did not have an effect. And we weren't surprised because this was not a strain that could grow inside the hive. But the big takeaway from this was that we were able to show that it can be spread throughout the hive. And so when the Metahesium becomes available, we'll be able to use the bee vectoring system to deliver the Metahesium, as well as we plan on developing our own strains over the summer as well that are more likely to survive within a hive and be effective against Varroa mites. Another thing that we were looking at is using bee vectoring for probiotics. So probiotics is relatively new in terms of being used inside hives. And there have been some good studies showing that, that if you disrupt the gut biome of bees, for example, after you use antibiotics, that can actually affect their mortality as well as their development and susceptibility to diseases. So repopulating those microbes can be very helpful, and those microbes are found also within the comb itself as well, within the hive, not just the bees. So repopulating that can actually be very healthy. In fact, there's a company called Strong Microbials that make a product called Super DFM, and a lot of commercial beekeepers use it after their bees have been really stressed out with pollination, being taken out to pollination. So we've done some preliminary studies with that. The bees are very happy to carry in the probiotics into the hive. And so we're looking at possibly using the probiotics with the bee vectoring this coming summer. And because it's already a product that is already in powder format and is actually considered a food product, it's something that we can actually use right away with our system, which is fantastic. Looking forward to other things that we can do with our system is the treatment of other pests and diseases. So small hive beetles are a huge issue for a lot of places. And so there's a lot of physical deterrents that are being used for small hive beetle, but potentially we can use these biocontrols or other things to manage small hive beetles and use that with bee vectoring. Nosema, wax moths, There's new mites and so forth that are coming in. So all these things are candidates for being treated with our bee vectoring system. Now the Protectobee, so it was the Inspencer, and then we asked for names, and Protectobee was voted as the most popular name. We took the Protectobee out to beekeepers. So we tested with 18 different beekeepers. We have 16 on here because we didn't get 2 of our surveys back, but we tested with about half of them were hobby beekeepers, about 24% commercial beekeepers, and 24% small scale beekeepers. And we had them put them on their hives, and then we basically said, hey, can you tell us, what you think about this for bee vectoring? Like we just planned on bee vectoring. That's what we were doing. And the beekeepers were thrilled. They loved it. In fact, when I went back each and every time to pick up the Protectobee, the beekeeper would say, can I please keep this? And I would say, why do you want to keep this? We don't even have the powders yet. And they would say, well, it's actually really nice. We were in our towards the end of summer here when the wasps get really bad. And so they were like, it's keeping the wasps out. Richard over in the far left here, he says, the skunks are staying away, and they just really liked it. So we realized at that point that we could use this for more than bee vectoring, and we could actually get it out to beekeepers, and that would get them used to the device, but also all the benefits of having the system that had these changeable inserts. So we've made the changeable inserts just simply so you could turn the cones around on either side, and that would allow you to direct traffic in and out on the right versus left, or the left versus right. So that had been why we had made it, but we didn't realize that the beekeepers were going to just basically fall in love with this thing. They loved it, and all of our beekeepers that have trialed it, they're so excited to get it now. So it's actually great. So we realized that we could protect from other predators, and that hadn't been the reason that we had done this, but we know why now that we've been able to reflect why it would protect against other predators. So we picked the color red because bees don't see red. And by having it so that the bees could not see red, that means that they weren't going to try to go in through the out, right? They couldn't get on, they wouldn't look for those edges and try to get in. So they would just see kind of this gradient where they were supposed to go in. And, they orient to the shape. They also have the smell. And when we first put it on, there'll be some fanning as well. So they figure out the in and out, but they're not trying to get in through the out. So that was why we originally did red. But it turns out wasps and other insects also don't see red. And so now the wasps don't actually, or yellow jackets or hornets or, all of those things, they aren't seeing it. So they don't actually know where to go in. It's farther away from the food source. And in fact, robbing bees, it's a little harder for them to get in too, because there's now these only 8 entrances to be protected as opposed to a big opening. And so, we had never expected that the cones would be put on all the time when we did it, because we were doing this for bee vectoring, but we had left the cones on over the summer, just as we were doing our projects and everything. And we found that the ventilation, we didn't have molding issues. We didn't have any issues with honey curing, and overall the hive health was the same as our control hives. So we think that the cones could be left on throughout a season. At least in our weather, it worked okay. If you're in the desert, I wouldn't necessarily want to leave them on all the time, but yeah. So so then we realized we had this protection for bees, like it was able to protect from other predators, and the beekeepers had told us about the skunks. And then they were like, well, do you think you can make entrance reducers? And we're like, well, yeah, we can easily make an entrance reducer. That's not a problem. So we've been testing the entrance reducers, and the bees don't propolis them. So it's really nice to take on and off. And then this fall, we put a solid insert. I should hold this up here. So this is the Protectobee here. And so we now have a solid insert, and then we have an entrance reducer that we put on the other side. And behind the solid insert, we're testing different types of feed. So if you need to feed your bees because it's cold, or you need emergency supply, or maybe there's a dearth, like it's not rained for a long time and you need to get some pollen in, by blocking it on one side, we can actually just slide in food there. And it's just like another easy way to go ahead and, take care of the bees. So this is my cool little 3D thing. So add powders or protect from robbers. The powders originally was what we were planning on. We had beekeepers say that they could turn it around, and then if they were weed whacking, they wouldn't have to worry about the bees coming out. And then easy entrance reducer. All my entrance reducers like are warped. It's really hard to get in, or they get propolized and I have to like lift up the hive. So this makes it a lot easier. And if you look at this with the open configuration with the maximum traffic, you actually have an extra large landing board. And so, it can block wind. That was just brought up the other day on a show that I was on. It can also, provide extra fanning area. And if you have any predators that are coming from above, then it can also protect from that. And in fact, the bees will sometimes, they'll beard underneath the Protectobee. So it also provides an extra level of protection. All right, so what's the future for us with the Protectobee? In terms of our timeline, we launched in February on Indiegogo. So we have, I think 10 days or so left on Indiegogo. And so we're using the funds that we're raising from Indiegogo first to actually pay for the tools and the products to make the Protectobee. And then the money then is going back into our research for this season. So we've got our first production run happening at the end of this month. And then we're delivering our product, we're hoping for June 2022 from the Indiegogo campaign. We'll still be selling later on. It's just those won't probably get to people right at the beginning of beekeeping season. It might be later or next year. In terms of our future plans, so the Protectobee production, we've got a bunch of additional inserts. People keep giving us lots of ideas of different inserts that we can use with the Protectobee, so we're going to be doing that. We're also going to be offering up a beekeeping mentorship that actually is coming now with the Protectobee. If you buy the Protectobee, you get a 1-year beekeeping mentorship. And this is an online mentorship where people will get notifications of when they need to do their treatments or check for mites or pull their supers or prepare for winter or what they need to do in spring. And this will be very regionally based. So we're building up a regional program so that it's specific for where you live, because we recognize there's huge differences within areas in terms of what you're doing during your beekeeping season and what your issues are for your beekeeping season. Small hive beetle is a big issue. And we don't actually have small hive beetle in our research apiaries, but they're around. So we're probably going to get them within the next year or two. But that means we're collaborating with beekeepers that are farther south to see if our product will work as is for small hive beetle. And then we've come up with some other ideas that we're going to test out. We're going to be continuing that fungal research, the probiotic test research, the feeds and supplements, and then working with other scientists that have come to us with other ideas of things that we can use for bee vectoring. So those are all the things that are coming up. We have a lot in our future plans that we're really looking forward to working on over the next few years. All right, move. So if you're interested in helping, we are looking for help in several ways. And so we've, as I've mentioned, I've always been this huge community person. I work with my community. When I had issues, I reached out to the beekeeping community. I think as in general, if we can all work together to solve our problems, that this is really the best way to do this. If we want to really save our bees and help our bees, working together is the key to getting to that point where we can have healthy bees. So the easy one, if you want to help our research, you can back our campaign on Indiegogo. We'll provide a link. You'll get an email tomorrow with a link. You can also just Google Indiegogo ProtectABEE and you'll find it. And those funds we're raising are going right back into research. We're also looking for citizen scientists, citizen beekeeper scientists. If you have the ProtectABEE and you're in different areas, we really are interested in finding out how things are working in your area, what the issues are in your area. We may be sending you things to test in your area. And so we're really hoping to collaborate with people in lots of different places so that we can, optimize this so that it works where people are doing their things. And as we're doing the beekeeping mentorship, that we know that we're giving the right advice for the people in those places. Are you located near us? I saw a lot of the Ontario people coming up when I asked at the beginning. So if unfortunately you come through the spring and your bees are alive, but they're very heavy with mites, we are happy to come take them from you. So please reach out because we're going to continue on with our mite research, and it's really hard for us to get mites early in the season. We usually have to wait till later in the season. So yeah, if you can contact us, that would be fantastic. Email and all of these is info@bestforbees.com. All right, to kind of wrap this up, I just want to thank my hive. So a lot of people say I'm the queen bee, and I'm like, oh no. Like we are all working together, and we're all working very hard. And I would never want to be the queen bee. That's a hard job. So anyway, so our research and development team, Nicole Gavreau, who's on here, Tasman Brown, Aparna Karthikeyan, and we have a couple of designers. So Amy Savigny and Victor Nobre are helping out with that. And then of course, Peter Kevan, who is the guy who with the idea, and he brought me into this whole thing. And again, we need to thank the people that have funded our research up to now. So MyTax is a government agency that supports universities working with industry. And this actually was started through the Weston Seeding Food Innovation Grant. That's how it all began. I was a member of the Accelerator Center, and they helped me with the startup company side of these as we've switched over to production. And then I have an advisory council of a bunch of amazing people that have guided us through this new phase of Best for Bees. So I'm going to leave the contact information up here, and Nicole is going to look at the questions, and she's going to let me know some of the questions that you guys have. So if you have any questions at all, you can put them in the chat right now, and then I'll do my best to answer them. We're going to spend maybe 10 minutes or so answering questions. If we don't get to your question, feel free to, again, email me at info@bestforbees.com. And then again, this will be recorded, and you guys will get a link to that at our website, bestforbees.com, or our Facebook page, Best for Bees. All right, so Nicole, what do we got?
Yeah, so the first one Daniel asks is, when will you have a product that fits an 8-frame hive? A timeline on that.
All right, so yeah, Daniel, so because we have those wings coming out on the edge of our bottom boards, we're right now with our 10 frame, we have a really tight fit because we want to make sure that if this is going to work for small hive beetle, that they can't get in. And if you noticed in that video too, where the bees were trying to get in, so we've really tried to make a nice tight fit for lots of reasons. And that means that unless you have nothing, no wings coming out on your eight frame bottom board, it won't fit in there. So what that requires is an entirely new Protectobee. And so we're looking at probably doing that. It just kind of depends on our funding. We have a lot of people that have 8 frames that have asked for it. I'm talking to this Australian group tonight, and apparently that's what they run in Australia, is 8 frame hives as well. So it just kind of depends on our funding in terms of what we get, and it's just a funding thing. It's about $20,000 for us to make a tool to make the 8 frame one. So it just depends on what we bring in. So hopefully soon, Daniel, hopefully soon.
Awesome. Chris is wondering if there's a device to be added to prevent swarming, similar to the Queen Guard that you could typically use. Yeah.
So in general, if your bees want to swarm, they're not happy, they've run out of room. And I say not happy, they're actually very happy. They're ready to, go out in the world and make another hive, but they might have run out of room. So we don't want to permanently prevent swarming. One, because you would actually keep your drones in there, and the drones need to get out. They actually need to die. Otherwise, you're just gonna have a lot of drones eating your honey, and they also need to mate for genetic diversity. But we are going to make a swarm preventer, which is basically a queen excluder that you put at the front, that you can use, if you need to get new equipment, or you don't have enough time to get it, over a couple of days, but not as a permanent thing. So, and I should mention the cones allow, they're big enough for the drones to easily go in and out. The pollen doesn't fall off. Dead bees can be taken out. On that Indiegogo page is actually a video showing a dead bee taken out. So, they're big enough that a queen can get in and out as well.
Awesome. We have someone wondering if there's a potential relationship with OMAFRA or the Canadian government and their farmers, if they're planning on using the vectoring because they're using pesticides and things like that. So is there anything brewing with research alongside OMAFRA or the Canadian government?
So I think and that would be in terms of, I don't know if we don't do the bee vectoring out to crops, right? So that's not part of what we're doing with our project at all. And in fact, I haven't even tested whether the ProtectaBEE could be used for vectoring to crops. And the nice thing about what's in existence for vectoring to crops is that it's continuous. So it's, it keeps going out over and over and over again. So in our system, they usually clear it out pretty quickly. So I'm not sure if it would be great for vectoring to crops. And I do not know if there's a collaboration between OMAFRA and bee vectoring. There is a company in Mississauga called Bee Vectoring Technologies. Mississauga is a city near me that's in Ontario that Dr. Kevan actually got started, and his patent is what got them started for vectoring out to crops. And they would probably be a good contact to find out if there is a relationship between OMAFRA and farmers using that system.
Awesome. We also have someone wondering about upper entrances and how, if the device plays a role within that, or how you guys have thought about I guess some people use upper.
Yeah. So in terms of the bee vectoring, you can definitely have an upper entrance and the Protectobee on. They're not carrying the powder up and out. We have had upper entrances when we don't usually, but we have had them on, usually by accident when we've run our bee vectoring experiments. We've done that a couple of times and they haven't brought them out. So we have trays that look at that ahead of time. And then those openings are pretty small. So if you were using it just for protection, I think you could probably still use an upper entrance because they're smaller and the bees can guard that a little bit better. But it would really depend on what you're protecting against. Yeah.
So a couple people are wondering if you could just remind people when the delivery of the first units could be expected, or when that timeline. And maybe talk about a little bit of the cost of the Protectobee and how much and what people could be looking at for pricing.
Okay, so right now we're aiming for June for our first deliveries, and we're actually spending a little bit more on shipping. We've included shipping in the price, but we're going to spend twice as much on shipping to get it air freighted because cargo containers are taking a very long time to get here. Our first production is happening in China, but we are currently sourcing a domestic supplier. It just wasn't going to be able to get out to beekeepers for this beekeeping season if we didn't do it overseas. And then in terms of costs, right now, if you go to our Indiegogo, we have an 8-pack, which is $37.50 per Protectobee if you get the 8-pack. The 2-pack is $100 for 2, so $50 each. And then the price changes on the one all the time because it depends on time. Like as we get closer to the date, the price keeps going up. So I think are we at $59? I think for a single right now. So those are the current prices, and they include shipping, and that's US dollars. So that's how Indiegogo works. So those are US dollars for all of our Canadian people. Sorry.
Anne is looking for some clarification. She's wondering if it's the vectoring that encourages them to move throughout the hive and deters the bees from simply pushing the powder out.
So Anna, one of the things we have not revealed everything that's happening inside the Protectobee when you've seen any of our advertisements. And so there's multiple levels of technology that keep the bees from moving the powder outside of the hive. So it's the powder sticks on the bees if they walk through and they're foraging, but we always have bees inside that are cleaning. And so we kind of had to figure out how to maximize it for foragers going through and minimizing it for cleaning by those cleaning bees. And I know that might be a little vague in its answer. Once we get the Protectobees out to people, you'll have a little bit better idea of what I mean by that. But because we wanted to protect our intellectual property for a little bit before we actually get the first devices to people, we haven't let everybody know everything that's happening with the Protectobee.
Right. We have Ray asking if the bees ever have a preference for the entrance cones on the left or on the right side.
Yeah, so they absolutely do. And it changes throughout the season. So I think it just depends on how they're filling it up during the season. So what I do before I put the cones on is I'll watch the hive and I'll see where the activity is happening. And so because we're looking at vectoring in, we're looking at where the bees are coming in, and then that's where we put our in cones, and then we'll put the out cones on the other side. And on the same hive, that can be different the next time we go. So it's not like, oh, hive 8 is always doing left side and hive 10 is always doing right side. It changes. So we just watch and see what they're doing.
Great. I think that looks like most of the questions, or if not all of them, for tonight.
Yeah. All right.
Yeah.
Okay. All right. Well, thank you everyone for coming. And it's been a real pleasure. This is my first big webinar